We've been spending a lot of time talking about Upshot. I'm curious, Dr. Barnes, what your like if you do a risk analysis of the company between now and your next like big important milestone, what could go wrong? I mean, yeah, she laughs because like a lot.
All of the things that can go wrong can go wrong. Yeah, there's a large number of them. Um, I think LNPs are no different. The RNA is no different. You can have timing delays. You can have manufacturing snags. You can have translation issues. Ultimately, whatever you've seen, you get a different result in people. It's drug development.
Yeah.
It's it's a low probability business from where you start to the chances that you'll get an approval. Right, you have to start so many threads so that one of them is going to make it all the way through. But you try and shorten your odds by getting the right kind of proof points in animals in vitro ahead of time. You shorten your odds by using a known biology. Yeah, there's many things that can go.
Yeah, I probably should have qualified the question a little more. Which I mean,
Liked it.
oh, it's a good question. I mean,
It's a great question.
with a lot of
We
different
don't know
answers.
enough about it, that's for sure.
Yeah. If I were to narrow that question, though, I probably would have asked like organizationally, structurally, in terms of people, talent, manufacturing capacity, like assuming the science goes as well as science does during drug development. What that's within your control as the captain of the ship are you potentially anticipating a challenge around?
Well, there's a lot of good companies competing for talent. So everything that happens is done by the people in the company. So you have to be good at, I mean, as a group, as a company, as attracting talent. And when people are here, it's got to be an environment where they like to stay. They can always leave. The best retention tool that you have people not want to leave, not, golden handcuffs or other sort of perks. Just create an environment where people feel seen, heard, appreciated, where they feel they participate, they understand what's going on. Something I've always striven for is transparency as much as I can, so that everybody has a common sense of what we're doing, where we are, what's going on. I mean, obviously there are limits, but as much as I can. And I think that tends to help people buy into the mission. One of the things that people often say after they've interviewed here is that everybody said exactly the same thing in terms of what we're doing, what the mission is. There's no duality or multiplicity of views because we all have the same, we have a common view. So I think that's, people talk about culture as being important. I think it often comes across sounding very platitudinous, but I think culture is very important in terms of retaining people, because when you're in a good one, you know it. And then, things do go awry or sideways from time to time. And that is when the true value culture emerges, because that's if like, well, I'm here because, the pay's good and we're on a ride, I'm waiting for my stock to vest. And then it goes south. It's like, oh, my stock isn't worth anything. Like, well, everybody's out the door. I mean, if it's a place that people don't want to be at, but if it's a place that people do want to be at, they say, well, you know what? I'm going to stay. I'm going to see this through. And that I think is the real value of having a place where people like to work.
Yeah. You mentioned it's a competitive space to find talent. Is that more specifically so when you're dealing with circular RNA, or are there transferable skills perhaps from people who've worked in other labs and other modalities?
Oh, sure. There's plenty of transferable skills. RNA is still hot, so it still, I think, attracts talent. Circular RNA, perhaps a little better than another regular linear RNA company, unless it's an editing company. I think editing still has cachet in that way. But yes, it would be it'd be different if we were doing a new kind of small molecule, a new kind of chemistry, or it's an old kind of chemistry, but a new kind of target that we've chosen here to take biological risk because, hey, we found new targets. That's what we're going after. That's the hypothesis we're going to test when we get to the clinic. That is, I think, I think it's just, unless you're good at telling the story or there's something really compelling, that tends to inspire a little bit less readily than, gee whiz technology.
Yeah. Yeah. I'm going to shift gears here a little bit. Time flies when you're having fun.
I know.
We are going to run short on time here. So I'm going to shift gears. I'd like to give you an opportunity to talk a little bit about your partnership strategy. I think you mentioned you found solid support from pharma, biopharma companies, and you've got specifically partnerships with Merck and Renegade Therapeutics, maybe others. So share a little bit about those partnerships and what sort of your strategy as the leader at Orna has been in establishing those partnerships.
Yeah, so the strategy, I think, that noise is not coming through. The strategy really derives from the fact that we have a true platform. People like to say they have platforms, and investors like
Everybody says they have a platform.
Investors like to invest in platform because it means if the first thing doesn't work, you've got a second thing. The alternative to a platform is a single asset play. But it turns out that that's not a binary thing. Platforms, or at least an approach which give 2 or 3 products, okay, that's most things. It's less common to have a platform that could really do a vast number. Editing was one, certainly, because a lot of the liver targets are all picked over. But in the early days, it was all there. It was all there to be done. And it's almost like this embarrassment of riches of the number of things you can do. And the same is true for RNA, for our approach. We have fewer people than Moderna, but we are doing fundamentally the same thing as Moderna. We are putting a long coding RNA into a particle and administering it IV. That's what they're doing. Our RNA has a different shape, and we have other advantages we think that we can do it. And their delivery is different than our delivery, and that's a fundamental thing. It's not the topology of the payload matters, lines versus circles. I argue circles are better. But they're not necessarily the killer app back here. I think the real killer app is the delivery. We were the first to show immunotropic lipids. Now everyone's looking for them and trying to replicate what we've shown. But until we showed it was possible, no one was really doing that. So in terms of partnerships, then, you look across all the things you could do, and there's like, oh my God, there's so many things that you can do, and you can't do it all. I think the mistake a lot of companies do is say, mine, we're going to do it all. But one of the things I like to say, I like to challenge people is, how many products does it take to make a successful biotech company? It's just one. Mm-hmm. So, how many of these things do you really need? So it kind of defines the partnering strategy. We picked the area, we declared we're going for in vivo CAR. That's what we're going to do. And we will, this doesn't mean the partnering's off the table, but the kind of partnership we're looking for is going to be very different, more selective, narrower. And everything else that we can do, we'll partner that. We'll partner that out. We're opening to partnering that out. And so that is the basis of the Merck relationship, which is primarily focused on infectious disease. So we weren't going to do anything in that space. Particularly, we were a little bit late to the party. But we even before the pandemic, we were founded not to do vaccines. We were founded to pursue in vivo CAR. That's what we're doing. Now, the relationship with Merck goes a little bit beyond infectious disease as well. I think they have had relationships with other linear RNA companies, and so that they have an interest in the space. So it goes a little bit beyond that. In the case of Renegade was a company, also came from the same stable. It's another MPM company, was founded about 2 years after us, and it was going to address the delivery question. So it's going to be delivery focused, not payload focused. It was going to be relatively payload agnostic. Mm-hmm. And so, we thought, well, okay, we could always use some cool new delivery. So that was the
Couldn't we all?
that was the relationship with Renegade as they were building things out. Now, of course, one of the places they started to see is, could they get a better vaccine lipid? And so the relationship with Merck came along at a time when we already had some early hits out of that. And so that the Merck deal is actually a 3-way deal between Warner, Merck, and Renegade. Mm-hmm. But yeah, we continue to work with Renegade on other things. So there's another deal with a Chinese company called Simnova, which is a member of the Simcia family. And that was an opportunity to, from our perspective, firstly doing excellent work there, discovering novel binders, novel CAR proteins, which they're clinically validating themselves. So it was a way to potentially get access to that, but also to get access to accelerated clinical evaluation through a particular setup that they have in China called investigator-initiated trials. It's just, it's something that most Chinese companies do, and something that we could do with them in our collaboration. So that was appealing to us as well. I like to think we were already thinking globally, but actually there was there was a practical angle to that.
Yeah. What about clinical opportunities in Australia? That seems to be all the rage right now.
Absolutely. In terms of not so much the investigator-initiator, in terms of formal evaluation, the Australian environment is obviously it's a top-notch medical society, medical healthcare society. It has typically simpler review processes, shorter times, and there's a particular interest in drawing RNA investment into Australia by governmental agencies. So there are various financial incentives of going down there as well.
Yeah.
So yeah.
It's positioning itself quite nicely as a hub over the past couple of years. It seems like there's a lot of really exciting mRNA stuff going on in Australia.
Well, we don't want to hype it too much. We drew Dr. Barnes to Boston. We don't want to lose him to to Sydney. And in the time that we have left, let's talk about where the space is going.
I've been somewhat obsessed, so I'm gonna selfishly ask the next question because I'm really curious to hear your answer. I feel like I might already know it, just given your love of RNA, circular RNA. But I'd really love to I often am talking about the RNA toolbox, the identity proposition that the different modalities can play within the broader biologic space, even just within the mRNA sector as a whole, right? Moving from, I think the space in general is moving towards some of these next gen modalities more regularly away from the linear mRNA for a number of different reasons. But I'm curious to hear, how you foresee circular RNA really fitting into or perhaps dominating. Do you see it fitting into more as a one tool, right, in the toolbox, applicable, kind of only in very specific use cases, or do you see it being used in partnership with a lot of different mRNA therapies and biologics, or do you just kind of see it dominating the space, right? Does it become the one to throw in a Lord of the Rings joke, the one ring to rule them all? I mean, you're welcome to steal that as well, by the way, but we might get in trouble with the Tolkien
copyright.
estate.
Yeah. It's an interesting question. So right now there are certain IP barriers to getting into circular RNA, but in the academic world, of course, that they're free to do research. And I think there's a lot of circular research going on right now using the method that Alex and Dan invented because it's just so easy and they don't have a constraint that way. So there's going to definitely be ongoing research and with circles or what you can do with circles. The main limitation, though, the thing that will limit its reach is how many different delivery solutions you can. If all you have is liver delivery, it doesn't really matter at the end of the day whether it's a circle or line. You might get slightly different performance, but what is really going to push things forward is the delivery question. And at that point, if you were forced to suffer with a mere linear RNA, but you had a very, very cool new delivery, you can make fine products with that, and nothing's prevented in that way. Mind you, if you had the circle, it would just be easier and better. So I think people are going to continue studying natural circles. That still is a bit of a mystery what they're doing there. I think people are going to do some technical innovations. An area where we've been focused technically on the circle side is how you get expression, because there is no cap, there's no end to the RNA. So you have to mark the start of the protein with a different sequence element. Now single-stranded RNA viruses use a trick to do that, and that's where we're drawing our inspiration from. But we've been doing, I think we've been leading in that space. We've been the first to really explore that in more of an omics way. And so we're really trying to define the universe of possible ways of expressing, at least as already instantiated in nature. Obviously, you could go to random sequences, but amongst those that are out there, and there's a lot that are out there, so it is a little bit daunting. And we use, omics approaches, we use machine learning to get after all that. So I think, notwithstanding the IP barriers, I think there will continue to be a lot of interest in circles, and there'll be a lot of people trying to, work their way around those patents in some way or other. Maybe they'll just be straight up very clever ideas that are very different to the ones that we have that get you there. And once you have the circle, you will enjoy all those advantages. Now, of course, you still have to express. And so that's why we think the expression system might be another line of defense. I know you're trying to think about opening doors, and here I am talking about closing doors.
I think that's an important conversation to have, to be honest. You need to talk about some of the potential door closers as well, right? I mean, this is new, and there's
we want to protect it. I mean, that's where our value is. But when you look across the modalities, and there are companies now doing tRNA. And the idea is that it's basically nonsense suppression, stop codon suppression. So all those genetic diseases which have a premature termination codon, maybe one therapy. It's a very intriguing idea, and so on. So there's going to be continuing approaches with RNA. Not, I think, less about circles versus lines, but creativity will emerge in other ways. So tRNA, or we see RNA editing as something that's out there now, or there'll be other kinds of RNA splicing things that are going on. I mean, so there's, there'll be RNA this and RNA that for sure.
Okay. Yeah. Whole universe of opportunities that will work in parallel, most likely to each other, right? Not just, one ring to rule them all, right? One solution.
Not if Dr. Barnes has us. Dr. Barnes, 2020 was an interesting time to start a biotech company. What do you wish you knew then that you know now? And whether that answer is specific to just biotech leadership in general, or specific to the RNA space.
So I'm a first-time CEO. So back when I was starting, I had to at least have the courage of my convictions that I could do a halfway decent job at it. I learned what it really feels like being a CEO. I'm not sure I would, just get ready for the ride is maybe what I tell myself, but I'm not really sure my younger self would have even understood, it'll be like this. Yeah, I think you have to sort of live through it to really sort of sense. It's a very central role, I think, at the CEO level, particularly in the early days. You basically have to do a lot of different things because you don't have the team is not fully fledged. But then, as it grows, then what you're trying to do shifts and evolves and changes. And, there's raising money. Is the story good enough? How do polishing your craft in terms of pitching and getting people interested in what you're doing, getting the message across succinctly or in an exciting way. And there's your board as well, and making sure everything is very smooth and aligned there. I've been in any number of board meetings before as a participant, but not as CEO, which is a far more central function in that. So that was maybe the newest thing to me, because I'd done early stage startups before where I didn't have the title CEO. I was basically doing everything anyway. So the early phases of Orna were very familiar. Yes, I don't know that there's advice. Just to say that it's a lot. It's a big role.
Yeah, there's advice in all of it. I ask questions like that all the time of my guests, specifically like scientist turned CEO, and the answers are wildly varying, but there's meat in all of them, right? Like someone's going to take something away from it. So just one follow up to that, and then we'll let you get on with your day. You said get ready for the ride. Looking back, what aspects of the ride were you perhaps the least prepared for?
Um, well, we went through some interesting macro factors. I mean, I've been in biotech long enough. I've seen various macro factors as an employee. Things go up, things go down, but it's other people's responsibility in some ways to deal with it or ensure the health of the company. And you know, there was the big boom time in 21, and then it all sort of went south, and sort of the investment dried up. So that was, that's really out of your control. And so you're presented with a scenario, and you have to solve for what you're trying to solve for. And then if the conditions change, you have to resolve for that. So that's less, that's not you. I mean, that's your team at that point. That's everybody from your board to your executive team to yourself, and then everybody in the company who's trying to create value through what they do. I mean, the path to salvation is value creation. I didn't mean that for that to rhyme, but it did.
You're a poet.
And I don't know it.
Well, that's, the conversation's coming full circle.
Oh my God.
Do you see what I did
Oh
there?
my God. That just
So anyway, that's the ride, I would say, in this particular case. It could have been a very smooth, a steady period in biotech over this last little while, but it's definitely been up and down.
Yeah. Yeah.
prepare to wear many hats, in other words, right?
Well, I could selfishly continue this conversation for quite some time because I've enjoyed it thoroughly. I think co-hosting's fun.
It is fun.
Yeah,
We're
we're
gonna
gonna
do
do more of
this
this.
more.
Yeah, RNA. I'm gonna find RNA companies so that I can just
RNA companies, pitch Matt.
do the light lifting and
You'll
just look
get
at
this.
things that
You'll
you
get this. Yeah.
Dr. Barnes, we really appreciate coming on. Thank you for joining us.
Thank you. Thank you.
Thanks very much for having me.
Yeah.
It was a lot of fun. Thanks, Matt. Thanks, Anna Rose.
We will we'll revisit maybe down the road when there's another inflection point to talk about. We'll catch up with Orna. But in the meantime, thanks again.